Hello everyone, welcome to part 2 of Recombinant DNA and Biotechnology lecture. So in this second part, we are going to now discuss the DNA sequencing technique, determining the gene function, discuss the application of stem cells for different medical as well as biotechnology purposes, application of DNA technology, and then finally discuss the safety and ethics of DNA technology. So for most of these reactions that I've just been explaining to you in the first part of this lecture, we were just discussing how if we have a gene of interest and we know its sequence so that we will create a probe that would flank the gene of interest. Now here in this second part, we are going to discuss how we know the DNA sequence, how we can use different techniques in order to determine the DNA sequence. And this is basically the principle of Human DNA Genome Project, which has discovered the entire sequence of the human DNA. So basically for DNA sequences, there can be several procedures that can be used. We're just going to discuss the traditional technique, which is the chain termination method. And I'm going to go over that in a second. But basically, in order for this reaction to happen, what we need is that we will need a single stranded DNA molecule. We need a short primer to start with, and this short primer will be complementary to our single stranded DNA. Then we will use a DNA polymerase and deoxyribonucleotide triphosphates, like for instance DNTPs for adenine, thymine, guanine, and cytosine. And then we also need dideoxyribonucleotides. And the difference between these two is that DNTPs on their 3' chain, they have a hydroxyl group, while DDNTPs or dideoxynucleotides, they lack the 3'. So they lack the 3' hydroxyl group. As a consequence of which, DNA, the nucleotides, cannot be added to the growing chain once DDNTP is incorporated. So let me show you how a phosphodiester bond can be formed. So here we have a DNA sequence that are attached to each other. Two nucleotides are attached by a phosphodiester bond. Now what polymerase does is that it catalyzes the formation of another phosphodiester bond through the 3' hydroxyl group. Now if this 3' hydroxyl group is missing, like for instance in nucleotides, in dideoxynucleotides, therefore DNA elongation in the PCR reaction can no longer happen. And so we can use this technology in order to find what's the DNA sequence. And I'm going to give you an example because that's the easiest way of showing you how the chain termination method works. So let's say we have a DNA sequence that its sequence is GACT, just for argument's sake, right? And we want to sequence this one. We don't know that its sequence is GATC. All we know is that we have a primer that can start behind the gene of interest. So here we have a 5' to 3' primer. So what we do is that we will amplify this gene using the PCR technique. So we have now the polymerase, we have the RNA primer, we have the template DNA. The only difference is that in addition to deoxyribonucleotides, we have dideoxyribonucleotides that we are adding to this reaction. And so anytime any one of these dideoxynucleotides get incorporated into the growing chain, the chain will terminate at that stage. So let's see what we will get right here. So we'll start to amplify. So here we have the sequence 5' to 3', and our primer here is in a 5' to 3' direction. One scenario is that the chain will be completely formed, so we get CTGA. But then the other scenario is that before this last nucleotide was added, one of these get incorporated there. So let's say that we get A. So that's one product that we got here. Now, what's the other scenario? We can have CT added, but then the third nucleotide is another DDNTP. So here, the DDNTPs, I'm going to show them with a star. And so the chain terminates right here. No more we can add right here because the 3' hydroxyl group is missing. Now the third scenario is that we get C and the next molecule is again another dideoxynucleotide. So now we can't add any more nucleotide right here. And then finally, the final scenario is that our very first nucleotide is actually a dideoxynucleotide, and so nothing more will be added. So now we got 4 different PCR products that their difference between them is only 1 nucleotide. And so based on this principle, we will use the difference in length by just 1 nucleotide in order to separate these PCR products from each other. And so there are different techniques that can be used. A fancy one is actually to use different color probes for different DDNTPs. So let's say that A uses the purple color, G emanates the green, T emanates blue, and then C emanates pink. And then there will be a machine that will detect as these products are running through. So these products are running through. The heavier one, the longer one will take longer to go through, and the shorter one will go faster. So the machine will tell us that, oh, the shorter one had a C, and then the one after had a T, and then the one after had a G, and then the one after had an A. So it gives us the DNA sequence of the chain that has been produced. And we know that the last one is on the 3' end, and the first one, the shortest one, is on the 5' end. Okay, so now we know that the sequence of our product is CTGA. So what was the sequence of our template? This was our template, and we didn't know at the beginning what it is. So if we want to know 5' to 3' direction, so 5' to 3' direction, TCAG. TCAG was the sequence of our original DNA that we have used by this technique. All right, so that's about it. That's the chain termination method. Here I have done a drawing for you to show you how, if we have the primer to extend our DNA, different DDNTPs keep getting incorporated, and so we get different sizes of fragments that can be run into a gel. The smaller fragments will run through the detector first, and the larger ones in the end. So basically this technique will help with the sequencing based on the difference in the length by just 1 nucleotide between every one of these DNA fragments. All right, so 1 nucleotide difference is between every one of these DNA fragments. Now that's a kind of like a more fancier technique of using a detector with an excitation laser by having different fluoresceins attached to these DDNTPs. Now we can talk about the more conventional technique, which just uses the agarose gel to isolate them. So here, let's say that we run the same PCR reaction, but instead of mixing all the DDNTPs, we run it in 4 different reactions. So one reaction, we will only provide DDATP. So all these things, DNA template, polymerase, nucleotides, and primer are present, but we are going to run 4 different reactions. In one reaction, we also add DDATP. In the second reaction, we add DDGTP, DDCCTP, and then finally in the 4th reaction, DDTTP. All right. So in the first reaction, all we get is 4 different fragments, because anywhere that the A will be incorporated, those are the fragments that we get. So we run it on the gel and we get 4 different fragments. Now same thing applies with the second lanes. Only the ones that have the G will be isolated. So we get 2 fragments right there. Then we have the third reaction, the ones that have the C. There we go, we get 3 fragments. And then finally, the one that had dideoxynucleotide for thymine is getting isolated. So 1, 2, and 3. And so if you pay attention, all these nucleotides are being separated based on the fact that there is only 1 nucleotide length difference between every one of them. And that's good enough. The resolution is high enough in order for us to get a right sequence. So, which of these DNA fragments is larger? So we always know that the top ones, they're larger because it takes longer for them to go through the agarose gel pores, while the shorter ones will run faster. So here we have the shorter ends, and so they're closer to the 5'. As we go to the higher end, then they have reached more to the 3' end of the DNA fragment. So here we have the 5' to 3' end sequence. And so our 5' to 3' end sequence is T, C, A, G, T, and so on. All right, so that's what we got there. And that's for the molecule that has been amplified. Now, in order to find out what was our template DNA, all we have to do is get the sequence from 5' to 3', which would be what? T, G, G, C, A, T, T, and so on. So here, 5' to 3', we get T, G, G, C, A, T, T, and so on. So that's the chain termination method by using dideoxynucleotide, which lacks the 3' hydroxyl group. And so anywhere that they bind, the PCR reaction will discontinue at that stage because no more nucleotide will be added after that. All right, now the next concept is the genomic sequencing. So for the first experiment we just discussed, we needed the sequence of one small gene. But what if we want to know the entire sequence of the genome? Like for instance, human genome, that was the genome project that we have discovered what's the exact DNA sequence of the entire human genome. How could that be done? And there are two different approaches that can be done for genomic sequences. So one is called the shotgun approach, and the other one is called clone contig, which stands for contiguous approach. And so for both of these techniques, what we do is that we isolate the DNA molecule, and then for the shotgun approach, we will digest it into many different small fragments, and then sequence these fragments, and then use a computer in order to build up the sequence. Right, so now we know the DNA sequence using the shotgun approach. Or we can use the clone contig approach, where we start by reading the DNA sequence for one small part of DNA. Then now we know what's the rest of the DNA sequence. So we create a new primer here and read some more. And then create a new primer and read more. Create a new primer, read more. What is the sequences until we finish from one end, then we try from the other end and go to the end of the DNA. And so this is basically called primer walking. Basically, we'll have to find out what's the DNA sequence of downstream DNA in order to create a new primer, find out more sequence, create a new primer, find out more sequence, and create a new primer. So this is called primer walking, using the clone contig approach, and then in the end we will put all the data together to find out what is the DNA sequence of the entire genome. So that's about it about DNA sequencing and genomic sequences. The next topic is how to determine a gene function. So let's say we've isolated a gene and we want to know what is its function. So how could that be done? So the first technique is by homology searching. And so how that's done is that we have the genome for certain animals. Like for instance, we have the genome for Drosophila, and we also have the genome for human. Let's say that we isolate a gene in Drosophila that we find out its function is copper absorption. Then we use computer devices to see what gene in human has a similar sequence. And we find out that gene X, for instance, has 60% of its sequence is similar to the gene in Drosophila that is required for copper absorption. So this way we can have an idea that, oh, gene X is probably required for copper absorption in humans, but let's go and do some tests and find out if that's truly what it does. And so we've already discussed what we can do is that we can isolate this gene X, clone it into the flies, and see what happens. Does their body copper level all of a sudden increases? If it does, there we go. We've confirmed that gene X is required for copper absorption. So the first technique is by homology searching. And for that, we already need to know the function of one gene in one of the species that its sequence is known. Okay? The second technique is called bioinformatics. And basically, it uses the sequence of the gene in order to predict the positions of alpha helices and beta sheets in the encoded protein. So it's trying to find out where the alpha helices and beta sheets of the translated protein are located, and so this way it gives us some idea about the possible function of that protein that is being encoded by this gene. The next technique is called reverse genetics, where we will induce mutations in a gene via different mutagens. We can use compounds that can cause mutation, or expose the animals or the flies, for instance, to radiation to induce mutation in their DNA. And then find out, once we find a mutated gene, how the phenotype of that animal is affected. For instance, let's see that we mutate superoxide dismutase enzyme, which is required for defense against reactive oxygen species. Once we do that, the animal would be susceptible to 100% oxygen and will die at a faster rate. So that's one way, by knocking out that gene of interest and finding out how the phenotype is being affected. The next technique is called forward genetics. Forward, here, let's write it down. Forward genetics, where we will mutate the organism that is working with and look for a specific phenotype. Let's say we are looking for a phenotype that is susceptible to 100% oxygen. And now let's go and find out what gene is it that is mutated that has caused this phenotype. And then, for instance, we may find out that, oh, it's the catalase gene that has been mutated, as a consequence of which this organism is susceptible to hydrogen peroxide, which 100% oxygen can cause their death. All right, so that was different techniques of learning how different genes function. Now, next, I'd like to quickly point out different techniques that we can use to inactivate certain genes. Because let's say that, for instance, we have superoxide dismutase and we want to knock it out, see how it affects phenotypes of that animal, but we don't have time to go through all these mutations and then screen, find out, is the gene affected, is it not, and then by the time we find it, okay, now we can check the phenotype. Is there no direct method of knocking out this gene? Well, there are different techniques that can be used, and the first one is called, all right, so we are done with finding out the function of the gene. Now we are trying to find out how to inactivate specific genes. And two commonly used approaches I'm going to discuss right here. So here, let me use a different color. So the first one is called homologous recombination. And with this homologous recombination, what we do is that we allow recombination between a chromosome copy of the gene. So here we have our chromosome with the gene of interest, and then a second piece of DNA that shares some sequences similar to the original chromosome that we are working with. So here we have another one that shares similar DNA sequence. And once recombination happens, the gene of interest will be removed and replaced with some non-specific DNA. So that's one way of removing the gene of interest by homologous recombination. The next technique is called microRNAs. So 2 for microRNA or RNAi. You might have heard RNAi or RNA interference. All right, and what this technology does is that we are not going to knock out the gene directly, but any mRNA that is being produced is being targeted and destroyed. And so this way, since the proteins will no longer be formed, we can find out, okay, if we stop product of this gene from turning into proteins, what phenotype does the animal present with? So here, let's discuss with microRNA. So these are basically small single-stranded RNAs with a hairpin structure. So if you have a small RNA with a hairpin structure, they would loop on themselves and form a double-stranded RNA. And this double-stranded RNA will go and find its complementary mRNA sequences and cause its digestion. So if you have another mRNA sequence right here that has a complementary sequence to this single-stranded RNA, it will be destroyed. It will be cut at different places. And so therefore, since the mRNA cannot survive, there won't be any protein formation. And so now we can check the phenotype of this genetically modified organism and see how by application of microRNA or RNA interference, the phenotype has been affected. All right, so that's one other way of finding out the gene function. Moving on to the next topic, which is the stem cells. All right, now stem cells, these are actually undifferentiated cells of multicellular organisms that are capable of giving rise to indefinitely more cells of the same type. So you know some of our cells can only replicate for a certain number. These stem cells, just like the cancer cells, they can replicate indefinitely to produce more cells of the same type. And then some of these cells can undergo differentiation to give rise to different functional cells. So now let's discuss the difference between different types of stem cells that we have here. All right, the first one is the totipotent, also known as omnipotent stem cells, and these will give rise to all the cell types in the body in addition to extra embryonic and placental cells. So give rise to all the cells in body in addition to extra embryonic and placental cells. Next, we have the pluripotent cells, which gives rise to all the cells again, all the cells in the body, but they can no longer give rise to extra embryonic or placental cells. And so embryonic stem cells, for instance, are examples of pluripotent cells that can give rise to ectoderm, mesoderm, and endoderm that gives rise to different tissues in the body. And we have described that all these derivatives of ectoderm, mesoderm, and endoderm in the embryology videos of our MCAT ACE lectures. The next type of cells are multipotent stem cells, which develop into multiple cell types, but they are more restricted than pluripotent cells. So you know how pluripotent cells can develop into almost any cells like ectoderm, mesoderm, and endoderm, except for extraembryonic and placental. Multipotent cells can transform into many other cells, but not all of them. They're more restricted compared to the pluripotent cell. And I'm going to give you an example. For instance, hematopoietic stem cells, they can give rise to red blood cells, white blood cells, and platelets. But they can't give rise to the brain cells, right, or heart muscle cells, versus pluripotent cells can give rise to heart muscles or skeletal muscles via the mesoderm. And then some other definitions that I'd like you to know is called oligopotent, meaning that these stem cells can give rise to few cell type. And then finally we have unipotent, which can give rise only to one cell type. Now, what is the application of the stem cells? They can be used in genetic research. For instance, researchers have been interested in knowing why stem cells can replicate indefinitely, and so they found out that stem cells carry the gene called telomerase. So as our DNA replicates, every time it does, the end of the DNA cannot get replicated. And so it keeps on getting shorter and shorter until cells can no longer replicate. Cancer cells as well as stem cells have this specific enzyme called telomerase, which prevents shortening of the end of DNA. And so it allows them to replicate indefinitely. So stem cells can be used in different genetic researches. It could be used for their regenerative capacity, for instance, in order to help patients with different health problems like neurodegenerative disorders, cardiovascular disease, or diabetes. Let me give you an example. For instance, in patients who have age-related macular degeneration, or AMD, their retinal cells die at later ages. And so they start to have a scotoma at the center of their vision. It's like a black spot that they can't read. And so what researchers have started to do is they've placed some stem cells in the retina of these patients, and it has been shown that their vision has started to actually improve. So there has been some vision recovery. And then other applications is like, for instance, transplantation that has been used in leukemic patients and other hematopoietic disorders, and so it helps to produce healthy blood cells. But then what we need to be aware of is that these stem cells can replicate indefinitely, so there is a risk of cancer. And so once we apply any one of these technologies, like for instance this vision recovery example that I gave you, researchers are watching these patients closely to make sure they don't develop cancer or any tumor in their eyes. So there is a risk of cancer development by any application of the transplantation or regenerative capacity studies. Next, I'd like to describe how gene cloning and their application of DNA technology has been used in different fields. So the first one, as we just discussed, is protein purification. We've already discussed this extensively in our first lecture. For instance, we can clone the insulin gene and isolate the insulin protein and then apply it to type 1 diabetic patients for their treatments, because type 1 diabetic patients don't produce insulin. So now we have plenty of insulin protein that we could put it in the injectable form and help with the treatment of these patients. But then students sometimes ask, all right, so you have a bacteria, you place the plasmid in, and so this is the bacterial genome, and here's the plasmid, and it gives you insulin plus so many other proteins that are belonging to the bacteria, so many other proteins. How are you going to isolate specifically that insulin and use it for the treatment of humans as you are just describing? And so different techniques can be used for purification. So I'm not going to go through the cloning technique anymore. We already discussed how we can form a recombinant plasmid, and then with electroporation we place it inside bacteria and then increase those bacteria. So now we are having a culture with so many bacteria that are producing insulin along with other proteins. How can we now purify this protein of interest? So there are two different techniques that we can use. The first one is by adding a signal peptide to our gene of interest. So here we have our vector, and then we had our gene of interest right here. Now in addition, we can add a signal peptide right here that signal the protein in order to be exported out of the cell. And so what happens is that this gene of interest, once it produces the protein will be exported out inside the culture medium. And so now we know that all the protein that is found in the culture medium is containing that gene of interest product, which could be, for instance, insulin. So that's the first technique. The second technique is recovery of a fusion protein by affinity chromatography. Where we will do the same experiment, except that for our gene of interest right here, so this is our gene, and then we will add a fusion protein right there, and then product of the cells we will run it against gel chromatography. So here we have the gel chromatography. It has the stationary phase that has the adsorbents, and these are usually silica gels. And then we will load our protein extraction into the affinity chromatography. Now what happens here is that our gene of interest, so here is our gene of interest, will produce the protein, and then this protein will have a fusion protein attached to it. So here, let's say that we have the fusion protein, and these fusion proteins will bind to the silica gel right there. So here we have a silica gel in this column chromatography that will bind to the fusion protein. So once we load our protein, the ones that have the gene of interest will all be stuck in the silica gel, while the rest of the proteins that don't have this fusion signal will get excreted outside. Now that we have all the protein inside, in the next stage what we can do is that we can apply some solvent in order to release the protein. So now these proteins will be released, and now here we will isolate the elute, which contains the protein of interest. Now later on in this lecture, I'm going to discuss some of the ethical issues regarding the application of DNA technology. For human uses, I'd like to give an example that animals that are being genetically modified in order to produce these proteins, like for instance, if you use a sheep and you try to place a gene so that it increases the protein content of their milk, these animals will have high frequency, frequency of birth defects as well as premature death. So all these genetic manipulation in order to produce protein of interest can cause some problems with our transgenic animal. Alright, so that's one problem with the genetic manipulation of these animals we should be aware of. Alright, so we've already discussed production of protein was the first application of DNA cloning. Now next, what are the other advantages? Well, we could use gene cloning and DNA analysis in medicine. Like for instance, the first example we gave production of insulin. Well, insulin can be used for treatment of diabetes. Alright, so here, number one, we were talking about gene cloning and application of DNA technology for production of different proteins. This protein could be, for instance, dietary protein we are giving in sheep, or it could be insulin that is used for the treatment of diabetes, and that would have application in the medicine. And then another application in medicine is, for instance, producing genes that selectively kill cancer cells, and this is called suicide gene therapy. So you introduce genes that will selectively kill cancer cells. So that's another application of gene cloning. Gene cloning can also be used in agriculture. So number 3, agriculture. You can, for instance, add genes to plants in order to make them resistant to, for instance, viruses. Or add genes so that these plants will have their own insecticides, and so they would be resistant to different insects. Or you could do gene subtraction, right? So subtract gene, knock out certain genes so that you would decrease, for instance, the starch content of their products. And again, there would be ethical concerns regarding these changes, and that is, for instance, it's being told that transgenic plants can affect the surrounding environment and ecosystem. So that's again another ethical issues that we have to deal with once we are modifying these plants. And then finally, the last application of gene cloning and DNA analysis is in forensic science, where we can use DNA analysis, for instance, in order to find crime suspects. So we just discussed, if someone who has committed a crime leaves just a few amount of DNA in the crime scene, we can use PCR to amplify this DNA and make millions of copies of that. Now, just like our fingerprints is unique to every one of us has got our own DNA. There are certain areas of our genome that has certain number of repetition of DNA sequences. Like, let's say I have, for instance, 1,000 repetition of my DNA sequences. You may have 2,000. Another person may have only 80. All right. So we could amplify the gene of that suspect, amplify it by PCR, and then apply restriction enzymes. And so I would get, for instance, 4 DNA fragments. You would get 8 or 9 or whatever, depending on where the restriction enzyme cuts, and then the other person will get 2, for instance. So now you can go back and compare. This sample gave us like 2 restriction site cut that has resulted into 2 bands and gel electrophoresis. Now, of all the people that we have here, this person must be the one who has committed the crime, because it has the same pattern as the DNA that has been found on the crime scene. And this approach is referred to as DNA fingerprinting. So again, as our fingerprints is unique to every one of us, our DNA also have certain areas that is hypervariable in terms of their number of repetitive sequences. And so based on the different digestive patterns that they produce on the gel electrophoresis, we can tell which DNA belongs to which person. And then same approach can be used for studying DNA kinship. So let's say that my mother has a DNA fragment, like same DNA fragments that we would digest, that will yield such banding pattern on gel electrophoresis. And then my father will have another DNA fragment that would give this pattern. So if they are truly my parents, then my DNA pattern should be something like this. And so that's one way that we can find out that if a child belongs to their mother or father, or if they're adopted, or for any other reasons that we will have to do the DNA profiling. All right, now let's go over some of the ethical concerns with the DNA technology, and then finally we will conclude our lecture with some practice questions. So safety and ethics of DNA technology. The first problem is that early diagnosis of fatal genetic disorders may prompt the parents to terminate a pregnancy. So that's one issue we may be dealing with, because now we can genetically test the kid. So here we have a fetus, but we'll isolate the DNA, find out what is its genomic sequence, and we find out that, oh, there are gene mutations in gene A and gene B. And so this child will have some mental problem or whatever, and so the parents may decide to terminate pregnancy. To what extent can we alter the human genetic content? We could create superhumans that are extremely smart or are extremely strong, and so wouldn't that be concerning? Now, if a patient is diagnosed with a genetic disorder that causes severe consequences in progenies, should the spouse be notified of the results regardless of the wish of the patient? So that's again another issue that we may have to be dealing with. When should you decide to treat mentally incapacitated patients with invasive genetic techniques? And should we or should we not do that? That's again another problem. Will the high cost of gene therapy make it only available to wealthy patients? And then finally, can we allow patients to alter their traits like eye color, height, intelligence by therapy techniques? And what if they want to alter their intelligence or their body strength and so on? So what would we do in that case? All right, so these are some of the concerns about the DNA technology that is getting so advanced to make us closer and closer to reach these goals of altering and making superhumans. So now let's move on with some practice questions. So the first question is, a forensic specialist isolates 2 strands of DNA from a crime scene. After 4 cycles of PCR, how many strands of DNA will be available for analysis? And then the second question is, which of the following is a true statement regarding the following experiment? So go ahead, pause the video. Once you feel you're ready, you can run it and we'll go through the answers. All right, hopefully you had some time to go through the questions. So here we are starting with 2 strands of DNA, 2 strands of double-stranded DNA. And then we go through 4 cycles of PCR. So we already know each cycle of PCR will double the DNA. So if we are using 4 cycles, it's 2 to the power 4. So we started with 2 strands and we did 4 cycles, which multiplies the number by 16. So we get 32 strands. And if you'd like me to draw it for you here, we have 2 strands. Via the PCR, they will become now 4 strands. And then next stage, they become 8 strands. 2, 3, 4, 5, 6, 7, 8. So this is our first cycle, this is our second cycle, third cycle, and fourth cycle. So on our first cycle we had 4 strands, on our second cycle we had 8 strands, on our third cycle we will have 16 strands, and our fourth cycle we will have 32 strands. So 2 times 2 to the power 4, which is the 4th cycle that we got here, is equal to 32. Now, which of the following is a true statement regarding the following experiment? You inject a transformation vector into the somatic cells. What happens there? Now, in order to answer this question, I would like you to know the difference between somatic cells and germline cells. So germline cells are like sperm and eggs that are being produced from testicles and ovaries. These will be transmitted to the next generation. While somatic cells are like skin cells and so on, they won't be transferred to the next generation. So if you inject a transformation vector in the somatic cells, those cells will be affected, right? But it won't be transferred to the next generation. So let's see what happens here. A selected number of cells will express the transgene. Correct, a selected number of cells. The transgene will be passed to a selected number of cells. No, it's a somatic cells. It will not be passed to the next generation. All future progenies will be chimera. No. Our current progeny, we can call it a chimera because it has 2 different genetics. So the original cells and the transformed cells are different. But the future generation, since the genes are not being passed on, are not going to be chimera. A selected number of progenies will be chimera. Again, that is incorrect. None of them will be chimera. All right, let's move on to the last question. Which of the following is a true statement regarding the animal marked by the yellow color? So we inject a transformation vector, this time into the germline cells, and we made that with another sheep. What is true about this yellow color marked sheep? The transgenic sheep will be sterile. Not necessarily. It could be sterile, but not necessarily. So no, I don't like this answer choice because it could go either way. The recombinant gene does not pass on to the next generation. Well, it depends. If this animal has already picked it up, like from the germline cells, then all the cells are affected, including the germline cells, and it does pass on to the next generation. So again, this question cannot be correct, because if the gene has already been picked up, it will be transferred to the next generation. All cells express the same gene. Well, all the cells have the same DNA, because our genome is all the same, but not necessarily all the cells express the same gene. Let me give you an example. Our brain cells, the gene that they're expressing is way different than what our heart cells are expressing, and it's way different than what our kidney cells are expressing. So that's the whole point of differentiation of different cells, because they do different functions. So this is an incorrect statement. So far these were two incorrect. So all cells express the same gene? No, they have the same genes, but they express different genes. So that is incorrect. And then finally, all cells contain the same genome, which is correct, but express different genes. That is correct. So choice D would be the answer for this last question. So this is it for our second video of recombinant and DNA technology. We have described how we can sequence the DNA and as well as the genomic DNA using different approaches, shotgun and the contig approach. How we can determine the gene function by either mutations, by homology searching, bioinformatics, reverse genetics, or forward genetics we just discussed. We've discussed the stem cells, the difference between totipotent, pluripotent, and multipotent stem cells. We've discussed about the application of DNA technology, how it can be used for production of different proteins, how it can be used in medicine, agriculture, and for forensic evidences. And then finally, we've discussed the safety and ethics of application of DNA technology. Alright, so that would be it. Thank you all for watching. I hope you found this video helpful, and I will see you for the next video.